Hello everybody, my name is Sam Schmidt, a student in the Woldering Lab here at MSU. Today I want to talk to you about this classic paper, Highly Avid Magnetic Bead Capture, an Efficient Selection Method for de novo Protein Engineering Utilizing Yeast Surface Display by Ackerman et al. Found here. And so what the authors want to do is find a way to capture weak binders, so those in the micromolar affinity, from a large library using a small amount of antigen in a time-efficient manner. These are all things that fluorescent activated cell sorting historically struggles with, so the authors decide to look at a different way, using either an antigen-coated surface or antigen-coated beads. As we see here, the beads show a much higher enrichment ratio, so those were chosen going forward. The purity was then determined, and so we see that agarose beads have better purity than the initial population, but not as good as the magnetic beads. This is likely because the agarose beads' inherent porosity makes antigen presentation more difficult than on a magnetic bead. So the magnetic bead was chosen going forward for continued characterization. And so enrichment levels as a function of time are checked, and we see we get pretty close to maximum enrichment after about 2 hours, 2.5 hours, which is significantly faster than can be done in flow cytometry for a large library size, so 10 to the 8th, 10 to the 9th library cells in your library size. We also see that we maintain pretty good specificity after our magnetic bead sort campaign. And then finally, the authors look at enrichment as a function of valency, both of your antibody being expressed, so your expression levels, and as a function of your bead, so the lysozyme density. Unsurprisingly, as both of those increase, we get greater enrichment levels. Using this, the authors then come up with this protocol following where you start with your naive library, which may or may not contain binders, likely containing weak binders at best. So you do some negative selection on just a bead without antigen coding to get rid of any sticky yeast or things that bind only the bead, that would bind the bead regardless of antigen. So after you've removed those, you then do a positive selection with your multivalent target display, so your magnetic beads, and then you can repeat this with increasing stringency, following which you can do a sort to make sure none of your mutants have been truncated, and then do a standard fax enrichment campaign where you make mutations and then select for your improved binders, and repeat until you're happy. Finally, you can characterize them with sequencing and titration until you get your binder for your target of interest. So I highly recommend everybody read this paper.